I'm happy to present to you my poster for the ISMB ECCB 2023 conference entitled Mechanistic Insights into Vaccine-Induced Immune Responses Gained from a Data-Enriched Boolean Modeling Approach. So my poster here has the ID in 1924, and this work results from a collaboration between the IDMIT lab at CEA and the software company Dassault Systèmes. First, a quick introduction. So the title mentions immune responses. We will more specifically focus on the early inflammatory stages. Inflammations being complex multi-scale biological responses of a host to stimuli. So there are, as a matter of fact, increasing amounts of data available about those inflammatory processes that are classically exploited through differential and co-expression network analysis. However, both of those are limited in the fact that they lack, well, they are static first, and they lack mechanistic interpretability. Instead, we chose to work with dynamic Boolean models, which rely on a qualitative representation of a biological system, where biological entities are assigned nodes with two possible values, one for present or active nodes, 0 for absent or inactive ones. And interactions can be formalized as functions of logical operators in order to form a complex network. Here, for example, we have a small toy Boolean network with nodes x, y, and z, and the corresponding functions here. So why did we choose this particular formalism? Well, first, because Boolean networks can be represented as graphs to yield visual insights about the biological systems. Also, their dynamics rely on mechanistic information, which means that they are biologically relevant, and those variables being binary to perturb the system and to look at its output is both intuitive and interpretable. For example, here, a switch between 0 and 1 is easy to implement, and on the other hand, also easy to see and interpret. So now that we have established that Boolean modeling seems to be appropriate as a formalism to match our needs, we can ask ourselves, how do we build relevant Boolean models? Well, it is true that a large amount of biological knowledge is available in the literature or in more aggregated structured resources like pathways or disease maps, but the translation of that knowledge into simulable models is not always straightforward, and even when successful, the resulting networks are often incomplete, too generic, and they do not mimic very well experimental situations. This, however, can be overcome by integrating real experimental data into the process. And to do that, we conceived a two-step methodology. First, as mentioned above, we can build a network from prior knowledge by merging signaling pathway data, for example, adding data from protein-protein interaction databases, and cell types from cell type-specific differential gene expression databases. So this yields what we will call a naive network, so that hasn't seen any experimental data yet. On the other hand, we can take data from, an experiment, a study, clinical, preclinical, and pre-process it, which means basically clean it, apply statistical filtering, binarization, in order to have some exploitable data for a process that we call calibration, which is basically the choice of Boolean functions for the variables to match their respective measured experimental profiles. And we developed an in-house algorithm to do this that I will not describe within this poster here. So applying that methodology to the inflammatory response induced by the MVA vaccine in 3 animals, we obtained a large network of 202 nodes and 310 interactions. It can seem hard to read like that, as it appears on screen, but it can be efficiently studied with the dedicated software. For its dynamics, for example, we chose the differentially expressed cell type populations in the data as our output. And so we got here the following trajectory, with columns being the cell populations and rows the successive states of the system. As we can see, it reaches a steady state and a number of updates that matches the experimental data here, so H0 to H168. We observe a, very good correspondence between the simulated and experimental trajectories, with only the non-conventional monocytes and the TCD8 lymphocytes that show significant differences. Apart from this, we can see that in both trajectories, so experimental and simulated, we observe close expressions between lymphocytes on the one hand. So here we have B lymphocytes, TCD8, TCD4 lymphocytes, and T cells, and NK cells. And on the other hand, between monocytes, here classical monocytes, and granulocytes. Okay, and the huge advantage of the Boolean formalism is that because of its mechanistic sense, hints can actually be given as to what explains these common behaviors. We identified those to be the shared contribution of the CD14 gene for the monocytoid cell types, and that of the CD40 ligand, the CCL5 chemokine, and the AP1 transcription factor genes for lymphoid cell types. In both cases, the NF-kappa B inhibitors appear to be very important as well. And coming back to the non-conventional monocytes, which are here, and the TCD8 lymphocytes, which are here, we can see that they only have one parent node, so one arrow, whose simulated expression profile alone cannot explain the experimental cell type abundance variations. So, to conclude, we developed a workflow to integrate real experimental data into a prior knowledge-based Boolean network to model an MVA-induced inflammatory response in animals. We successfully built with that the Boolean network that best fits the experimental data, okay, given the fixed topology of the naive network, and that network carries mechanistic information about the causes for the observed outcome. Now, what's next? Well, it's to perturbate the network and validate the modified outcome experimentally. So thank you everyone that was involved in this project and everyone that watched this video. I'd be happy to get into more details if you want. So the poster has the ID 1924 and my contact information can be found on the bottom right side of it. Thank you. Bye.